And there we go. So we'll start off with the Pear Deck screen for just a minute. As I said, each Monday, 4 o'clock, we'll have these sessions. This is a blurb from your AP Biology formula sheet. Hopefully you guys have seen this and you're familiar with it. But if not, this will be on your formula sheet. You can use this throughout the whole exam. During today's session, we'll deal with everything you see in this little square. We'll talk about standard error of the mean. We'll talk about chi-square, and we'll use that chi-square table just a bit as well. All right, if you were here last session, we talked about chi-square most of the time last session. Today, we're going to quickly review that. We're going to talk about some things called p-values also, and then the rest of the session, we're going to deal with standard deviation, standard error of the mean, and how to create graphs with error bars and how to interpret those. But here we're gonna review chi-square first. Remember that chi-square is a type of hypothesis test that will allow you to test a hypothesis based on categorical data that's usually in the form of counts, counting things. Works really good in genetics. So here's a genetics problem. And as we're doing this problem, we'll be able to review some genetics, but we'll also be able to review chi-square. So I want you to switch back now with me to PowerPoint. So go back to your Zoom screen. And this way I'll be able to work the problem. Before you do that, if you will, go back to your Pear Deck screen for just a minute. And you've got the ability to write on the screen. So I want you to write, after you've read the problem, what you think a null hypothesis would be for this problem. So go back to your Pear Deck screen, read the problem, and then write out on the screen what you think the null hypothesis would be for this scenario. Take about 2 minutes to do that. All right, I've got about 23 of you with responses. So go ahead and I want you to write a null hypothesis for the scenario you see on the screen. Go ahead and take just a minute to get that done. All right, I'm now going to go back to my PowerPoint screen where I can write on the screen. And before I try to talk about the null hypothesis, I want to talk about the genetics of this problem a little bit. So it tells us that color blindness is a sex-linked trait. I wish it said it's an X-linked trait. Somebody unmute and tell me what that means for a trait to be sex-linked or X-linked. Anybody, just unmute and tell me what you think that means. Don't be bashful.
The gene is located on the sex chromosomes.
Right. So the gene for that trait is on, in this case, the X chromosome. And why that's important is that males and females are probably going to be affected differently by the trait. Also, when you're doing a Punnett Square for an X-linked trait, you're going to need to show the X and Y chromosomes in your Punnett Square. So this says that it's a sex-linked trait. The female is a carrier of the colorblind allele. So if the female is a carrier, what does that tell you about color blindness? Is it dominant or is it recessive? Somebody unmute and tell me. If there can be carriers for the trait, is it dominant or recessive?
Recessive.
It's recessive. So I'm going to write out some symbols here. So females, hopefully you know, are X, X. So there's the female. Here's the male, X and Y. So it says the female is a carrier for color blindness and the male is colorblind. Well, I'm going to use, I could use any symbols here, but I'm going to use big N to be normal vision, which means little n is going to be colorblind. And typically the way that you do this when you're dealing with an X-linked trait is you use these n's as superscripts on the sex chromosomes. So my female is a carrier. That means one of her X chromosomes has a normal vision gene. The other one, though, is carrying a colorblind gene. So that's my female. The male's colorblind. That means his X chromosome has the colorblind gene linked on it. The Y has nothing to do with this other than it's the other sex chromosome. It doesn't carry the allele. These are the genotypes for my two parents. So what I want to do first is I want to go ahead and do a Punnett Square quickly for this. So I'm going to, during meiosis, the two female X chromosomes separate. They go into different gametes. That's what I'm doing here. And then during meiosis, the X and the Y from the male separate. All right, one possible combination would be this. That's another carrier female, right? Someone unmute and tell me what we know about the person in the second box that I just filled in. What can we say about that individual?
Male and not colorblind.
Yeah, a male and normal vision, not colorblind, exactly. All right, then down here, that's a colorblind female. And then right here, that's a colorblind male. So essentially our Punnett Square is saying 1/4 of the offspring ought to be normal visioned females, that one. Another 1/4 ought to be normal vision males, that one. Another 1/4 ought to be colorblind males. And another 1/4 ought to be colorblind females. So those are our expected ratios. Now, before we go any further, remember what we're going to try to do now is we're going to try to compare the data that we got when we did this cross, this data, with the data we expected to get from this Punnett Square. Categorical data, counts. We want to compare them. That's what a chi-square does. Before we do that, we want to write out a null hypothesis. Anytime on the AP exam, you're asked to write out a null hypothesis. What's the fail-safe thing to write? What can you write and always get the null hypothesis question right? Somebody help me out. What would be the easiest thing to put here, no matter the question, and get a null hypothesis question right? Essentially, remind me, guys, what does null mean? You guys need to speak up a little bit. Null, what does it mean?
It's like none.
None. Okay. So when you're writing a null hypothesis, you're going to say that there's no difference between what? What are we comparing here?
The expected and observed amount of offspring.
Okay, yeah. But what I want you to say, though, is I want you to say that my null hypothesis is that there's no statistically significant differences between my observed data and my expected data. And if you write that, no matter what they're asking you for a null hypothesis about, you're going to get credit for it. So again, you're going to say there's no statistically significant difference between the observed data and the expected data. And if you say that, you're good to go on your null hypothesis. Now, to test that null hypothesis, we're going to run our chi-square. And the easiest way to do that is to fill out this table that you see on the screen. So we start off with a column of phenotypes. And I'm going to write in my different phenotypes. So I'm going to say female, and I'm going to use just a normal, female, and I'm going to say CB just as abbreviation for colorblind, since I don't have much space here. I've got males, normal, and males, colorblind. Now, since this trait is a sex-linked trait, we want to break this up by gender and trait. That's why I did females and males separately. If it wasn't a sex-linked trait, if it was autosomal, there would be no need to break it up by sex. But whenever you're doing a problem like this and it's X-linked, break up your phenotypes by trait and by gender. Next, we put in our observed data, and that's this. So normal females are 132, colorblind females 124, normal males 126, and colorblind males 136. All right, so how do we get our expected data from that? Somebody help me out.
You get like
Go ahead.
you like add up all of them and divide by 4.
Yep. So we're going to add up that observed column. I'm getting 518. You guys agree with my math there? Yep, 518. So I've got 518 total offspring. Let me move some things around on the screen here. So that's my total number of offspring. I know that I expected 1/4 of those to be normal females, 1/4 to be colorblind females, 1/4 normal males, and 1/4 colorblind males. So I'm going to multiply 518 by 1/4 or divide it by 4. And when I do, that's 129.5. And you go ahead and keep the decimal. And I'm expecting all of these to be 1/4 of 518. So in this case, all of these are going to be 129.5. And now to finish my chi-square analysis, I just do what the chart tells me to do. So I go O minus E. 132 minus 129.5 in that first row. So that's 2.5. 124 minus 129.5, negative 5.5. 126 minus 129.5 to get negative 3.5. And 136 minus 129.5 to get 6.5. All right, so now in the next column, I just do what the header says. I'm going to take this column and square it. So 2.5 squared, this is 6.25. 5.5 squared, and it's negative 5.5 squared, but negative times a negative is a positive. So I get 30.25. 3.5 squared in the next one, negative 3.5 squared to get 12.25. And then finally, 6.5 squared to get 42.25. All right, finish this up in the last column by dividing the previous column by E, by the expected value. So that would mean in my first row, it's going to be 6.25, this cell, divided by the expected value of 129.5. And that gives me 0.048. Always carry this out a couple more decimal places than what you have in your problem. You can always round it at the end. Next row is going to be 30.25 divided by 129.5. and that gives me 0.234. Next one's 12.25 divided by 129.5. And that gives me 0.095. And then finally, 42.25 divided by 129.5 to get 0.326. Now, I'm hoping that you guys remember that the equation for chi-square, chi looks like a fancy X, it's equal to the summation of O minus E squared over E. This is a formula off of your formula sheet. And really all that formula tells me to do is to do what from my table? What should I do here with my table to get my chi-square value? Anybody?
Add it up.
Yeah, add up what?
That last column.
Yep, add up the last column is all that formula is telling me to do. And I'm getting a chi-square value of 0.703. Now, by itself, that number doesn't really mean much. It doesn't tell you anything. You've got to compare it to what's called the critical value. And you get the critical value from this table that's given to you on your formula sheet. And the way you use that table is, first of all, you have to know how to find degrees of freedom. The degrees of freedom are going to be one less than the number of phenotypes you started with. So we started with 4 phenotypes, which means our degrees of freedom is 3. You're always going to use this 0.05 row unless they tell you otherwise. We'll talk more about p-values in a minute, but you're always going to use the 0.05 row, which puts us at a critical value of 7.82. So we had a chi-square value of 0.703, and we have a critical value from our table of 7.82. We compare those. So if our chi-square value is greater than our critical value, that's when we reject the null hypothesis. Well, in our case, our chi-square value is way less than our critical value. So our chi-square is less than our critical value, which means we're going to fail to reject the null hypothesis. Now remember, the null hypothesis is that there's no statistically significant difference between this column of data and this column of data. We're failing to reject that null hypothesis, which means that, yeah, our data is a little bit different. Our expected data is a little bit different than our observed values, but those differences are small and most likely simply due to what? When we reject the null, what are we saying the differences are caused by?
Chance.
Chance. On the other hand, if our chi-square value had been bigger than that critical value, we would have rejected the null hypothesis. And that would have told us that these two columns of data are very different, too different for the differences to be due to chance. That's really what chi-square is all about. All right, so that was a quick review of chi-square. Now we're going to move on a bit. And I want you guys to take a minute and read the blurb on this screen. So go ahead and stay on the Zoom screen with me for just a minute. Go ahead and read what you can see up on the screen, and go ahead and look at the data in the table at the bottom and see if you can make sense out of where we're going with this. All right, so one thing on these free response questions, make sure that you read very carefully. So we're looking at a type of plant H. squarosus, and we want to know essentially how an insecticide affects the plant. So there's some insects that eat the plants, and it tells you that in the problem. There's other insects that pollinate the plants. So we don't know which one this insecticide is going to affect necessarily. They do tell us that we take the plants in 10 plots, so just 10 pieces of land, and we spray all those plants in those 10 plots with the insecticide that's water soluble, meaning that it's dissolved in water. So what we're spraying is water and the insecticide. On the other hand, we've got 10 more plots that we spray with only water. And we'll talk about how crucial that is here in just a second. So the researchers recorded the numbers of plants or plant parts at each life stage in each test plot. And the results are shown down here at the bottom. So we're looking at the different stages of the plant's life cycle, when it's flowering, when it forms seeds, when those seeds mature, when they develop. And then we're left with essentially the density of the plants in the two plots. So this is the density of plants when only water spray was used. And this is the density when insecticide was used. Now, we need to talk about p-values here for a second. P-values use essentially a probability. When you're doing hypothesis testing, one way to do the hypothesis test is to look at the critical values and compare them to the value you calculate, like your chi-square value. And if your calculated value is bigger than the critical value, then you reject the null hypothesis. The other way to deal with hypothesis test is to get a p-value from a standard table. And that p-value, if the p-value that you're given, that you determine from the table, if the p-value is less than 0.05, that means that you're going to reject the null hypothesis. If the p-value is greater than 0.05, you're going to fail to reject. Now, here's what that means. If your p-value is less than 0.05, that means there's less than a 5% chance that the differences between your two groups are due to chance. If p is greater than 0.05, that means there's a 5% or greater chance that the differences are due to chance. And that's not what we want to, that's not where we want to go. So point being, notice that we have some p-values that are greater than 0.05 here, and we have some that are less than 0.05 here. Down here, these p-values are really tiny. That's telling us that the differences between these two groups of data are significant, where we're going to reject the null in these cases. Here are the p-values up at the top. These p-values are greater than 0.05. That's telling us that these two sets of data may be a little different, but the differences aren't great enough for us to reject the null. We'll come back to that in a minute. Let's go to the next part of the question. All right, I want you guys to go back to the Pear Deck screen with me for just a minute. And I want you to answer the first question, part A. Just write in your response. What's the control treatment or control group described in this experiment that we just read about? And then justify the researcher's decision to not use a set of unsprayed plants as a control. That word justify is probably the favorite word of AP question writers. Justify. So tell me what the control treatment is and why, essentially, is what justify means. Tell me why it was okay for the researcher to not use a set of unsprayed plants as the control. Quickly type in an answer, or write in an answer rather, for just question A is all you're answering right now. All right, so this part A is as easy as it looks like it ought to be. When it says identify, you're just going to say the control treatment was, and you guys are going to say, what's the control treatment in this experiment?
The plant spray with water.
Yeah, okay. Yes. One thing I want you to be real careful on, though, they want you to be really exact. Were the plants sprayed with insecticide also sprayed with water? Were the plants in the experimental group, the group that was sprayed with insecticide, did they also get sprayed with water? Yes or no? Somebody help me out. The plants that were sprayed with the insecticide was water soluble, which means it was dissolved in water. So they were sprayed with water. Point being, it's not enough just to say the plants that were sprayed with water. You need to say the plants that were sprayed with only water. So you got to be really exact with your words here. Say the control group was the plants that were sprayed with only water, no insecticide. Be exact with your wording. And the justification was, let's say you had a control group that wasn't sprayed with anything. Why would that not be a good control group? Why would a set of plants not sprayed at all not be a good control group in this case? Somebody unmute. Tell me why an unsprayed set of plants would not be a good control group.
It could probably affect like the growth of the plants.
Yeah, plants need water to grow, right? So if you sprayed some plants with water and others you didn't spray with anything, it might not be the insecticide causing the difference. It might be, well, some got water and some didn't. So you would be interjecting another variable, which in an experiment you really only want one independent variable, right? And in this case, that ought to be insecticide. Presence of insecticide or no insecticide. So point being, the only difference between my control group and my experimental group should be that one variable, the insecticide. I don't want water to be another difference. That's really what they wanted you to talk about in the justification part of part A. Let's go ahead and look at part B. It says, using the template provided, calculate the percent of plants or plant parts remaining from one life stage to the next life stage. Round your answer to one decimal place. They're always going to be that exact to the wording. So I'm going to go back to my PowerPoint screen now where I can write. And this is the table. This table was given to people on the AP exam. So we started off with 8,220 plants in the water spray group. And in the insecticide group, 9,490. So here, all of them are remaining, right? Because we haven't done anything yet. All right, but then as they age, a lot of them die. And when they enter the seed formation stage, only 2,440 of the original plants are still alive. So what are we going to do to fill in this spot right here? If we want to know the percent remaining from one life stage to the next, what do?
You divide it from the total.
Yeah, just go 2440 divided by 8220. 2440 divided by 8220. Now this asks us to give our answer as a percent rounded to one decimal place. So right now my calculator says 0.2968 with a bunch of other decimals. Well, first of all, I want it as a percent, which means I need to move that decimal 2 places to the right. So 29, and I want to round it to one decimal place. So 29.7% is what I'm looking at. I want to do the same thing in the insecticide spray column. So I have 3,100 out of 9,490. So I go 3,100 divided by 9490, and I get 32.7%. Now I drop down to the next row. What am I going to divide on the next row to fill in this box?
Divided from the previous stage.
Yeah, so it says compared to the previous stage. So I'm going to take 60 that I have here divided by what I had in the previous stage, 2440. So 60 divided by 2440. Again, I want to make this a percent. So I multiply by 100 and I get 2.5%. On the other column, I'm going to divide 1200 by 3100. And in this case, I get 38.7%. And then finally, the bottom row is going to be 1.4 divided by 60. So that's, again, 2.3%. And the last one, 33.1 divided by 1200. And that is 2.8%. All right, so if you compare column to column, obviously these are both the same. These are both pretty close, right? 32.7 and 29.7. But notice this column or this row here, 38.7 over here, 2.5 over there. And then these are very similar toward the end. So let's back up and ask us what it asks us to do next. Part C, based on your calculations, identify the plant life stage at which the insects have the greatest impact on the life cycle of the plant. And once again, there's that word, justify your answer. So where are the insects having their impact? Somebody unmute and tell me, where are the insects causing? Yeah, here. That's where there's way more plants alive in the insecticide group, than there were in the water spray group. So we could get it from here, or we could go back to our original table and see that that's where the p-value is really, really low. That means that the difference between the number of plants alive here and the number of plants alive there is significant. We couldn't reject the null hypothesis. We've got significant differences there. So our answer here would be the identify the life stage would be the seed maturation stage. And our justification could be that there's a much higher percent of plants still surviving in the insecticide spray group than there was in the water spray group. Or we could talk about the p-values and say that the differences in those survivors in the two groups is significant because of the low p-value, p less than 0.05. All right, on D, describe the most likely interaction between the insects and the plants. So in this example, what are the insects doing to the plants? Note that the group where we used an insecticide, more plants are alive than the group where we just use water. So that's telling you that what are the insects doing to the plants? They're eating it. They're feeding on the plants. They're killing the plants. So something to the effect of insects are feeding on the plants, they're killing the plants, something like that. Now that being said, now let's go to part E. In another experiment, researchers used a different insecticide. So different insecticide that affects a different group of insects. Make sure you read that closely. In this treatment, the number of seeds formed was reduced compared to the control. So they're saying that the number of seeds made in the insecticide group was less than in the water-only group. Based on this information, describe the most likely interaction between the affected insects and the plants in this experiment. So if the plants sprayed with an insecticide end up making less seeds, less babies than the plants sprayed with water only, what does that tell you those insects are doing? What are the insects we're doing in this problem that we're killing in this problem? What is their role for the plant? What I'm hoping you guys see that those insects are pollinators. And if we kill the pollinators, that means that reproduction doesn't happen and therefore seeds aren't formed. And that's what you want to talk about in the justification. So really you would say these insects are most likely pollinators because their role is to pollinate, to move pollen from male flower to female flower, and that causes the formation of seeds ultimately. Without the pollinators, seeds don't get made. All right, questions? All right, so let's go back to our Pear Deck screen now. All right, so for the rest of the day today, we're talking data analysis, which we've kind of already been talking about. But these are important things analyzing your data will do. They'll let you come to conclusions. You can make claims about your data. You can support arguments. You can look at the reliability of your data. And by analyzing your data, you can communicate those conclusions to other people. And we really talk about two kinds of statistics when we're dealing with data analysis. Descriptive statistics tell us some important characteristics of our data. When we're talking about descriptive statistics, we might be talking about things like the mean, the median, and the mode. Those tell us like the central tendencies. What's the middle of this data? We might look at standard deviation. Somebody tell me in one word what standard deviation really tells you about your data. What's standard deviation measure about your data? In one word. And that word is on the screen right now in the descriptive statistics section.
Like mean?
Oh, it looks at the mean, but it really tells you about the variability of your data. Variability is the word I'm looking for. How different are your data from each other? A set of data with a big standard deviation is all over the place. But a set of data with a small standard deviation, those values are very close together. On the other hand, we might also use what's called inferential statistics. And these let us take a sample of data and make predictions about the whole population, the whole big group, from a small sample size. We're going to be dealing mostly with inferential statistics for the rest of today. Here are some descriptive statistics that we might talk about: the mean, the size of the sample, the standard deviation, and the standard error. Now, this formula that you see on the screen right now is a formula for mean. This is on your AP formula sheet. Everybody in here I know knows how to calculate the average or the mean. But you might look at this formula and think, man, that looks crazy complicated. And it does. This X with the bar on top, that's the symbol for mean. All this says, though, these XI's are your individual data points. And all this summation symbol means is add up all your individual data points and then divide by n, which is the total number of data points. So this is just the normal average. It looks complicated. It's really not. Add up your data points, divide by the number of data points. That's all it means. Here's the formula for standard deviation. You will not be asked to calculate a standard deviation on an AP exam. Let me say that again. Even though this formula is on your formula sheet, They're not going to ask you to calculate a standard deviation. They may give you some standard deviations and ask you some questions about them, but they're not going to ask you to do the calculation. I'm going to calculate it for you here in a minute just to show you how it works. But remember that standard deviation is about variation. How different are your data points from each other? In a perfect world, you want a very small standard deviation. That tells you that your data is precise, that you're getting about the same thing each time you measure it. You can have a little bit more faith in data with a small standard deviation than you can with a standard deviation all over the place. And this is just showing you if we plotted the data with a low standard deviation are going to be really close to the mean. This dotted line represents the mean of the data. The data with a low standard deviation, all the measurements are really close to the mean. On the other hand, when we have a high standard deviation, the data are spread out, much like you see on the bottom, the big curve. It's all spread out, the one that's not tall. High standard deviation there, those data points are kind of all over the place. Think of that like a blindfolded person playing darts. That's kind of what you would get when you have a high standard deviation. On the other hand, a low standard deviation was when you have like a professional dart thrower. He's going to hit the bullseye or pretty close to it every time. He gets about the same thing every time he does it. That's low standard deviation. High standard deviation would be like that blindfolded guy throwing darts. You never know where those darts are going to be, and they're going to end up in a different place every time. Here we see the same kind of thing, just a different graph. So high standard deviation, data's kind of all over the place like you see on the left. Low standard deviation, the data is centered around the best fit line in this case. Very close to that best fit line. All right, we're going to talk a little bit about the standard error of the mean also. And again, this is a formula that's on your formula sheet. But again, you're not going to be asked to calculate a standard error of the mean. You're going to be asked to use it, but you're not going to be asked to calculate it. But standard error of the mean is equal to the standard deviation, which is S, over the square root of N. N is pretty much always the size of your sample. How many measurements did you take? So standard deviation divided by the square root of the sample size. And the way we're going to use standard error of the mean rather in AP biology is we're going to calculate that standard error of the mean, and it's going to allow us to estimate from our sample what the true population mean might be. And we'll talk more about that when we get to error bars here in just a second. All right. Give me a show of hands if you can do that in Zoom, if you know how to do that. If not, just type in the chat yes or no to this question. How many of you have been taught how to draw error bars on your graphs? So they give me like a thumbs up in Zoom, or just type yes or no in the chat room. I see a thumbs up there. See several thumbs up. That's good. All right, so the types of error bars that we're going to use are called inferential error bars. And an inference is like a prediction, right? So we're going to use them to make predictions. They're going to allow us to measure the mean of a true population, the big group, from a small sample. And we're going to use the standard error of the mean to create what are called 95% confidence intervals. And the way we're going to do that is we're going to, most of the time we're going to be doing bar graphs with this. Occasionally we might do some error bars on a line graph, but most of the time in biology it's going to be a bar graph. So you'll draw the mean on a bar graph, and I'll get to where I can draw on the screen in a second. And then you go up from that mean 2 standard errors, and you go down from that mean 2 standard errors. And you draw little horizontal bars. And then you connect those to the mean with a vertical bar. And again, I'll show you this in a second on the other screen. In between the top bar and the bottom bar, That's called a 95% confidence interval. And really what that means is that given the small sample mean that you have, you can now predict with 95% confidence that the mean of the whole population lies in between that top error bar and that bottom error bar. So from that small sample mean you have, you're now predicting that your population mean is somewhere in between the top error bar and the bottom error bar. So we're predicting a population mean from a sample mean. And we're going to talk more about that here on the next couple of slides. So another thing that you can use error bars for, and you can see some error bars on the screen, and I'm going to go back to my PowerPoint so I can write on the screen now. So you might want to go back to the Zoom screen where you can see this. So here we have plants, some plants that were grown, some ivy that was grown in the shade, and some ivy that was grown in the sun. And we graphed the mean width of the leaves. So the shady ones had a mean width of about 7.5 centimeters, and the ones grown in the sun had a mean width of about 6. And essentially, here we're making a claim that sunlight inhibits leaf growth, and therefore the width of ivy leaves. So essentially, we're saying that they grow better in the shade than they do in the sun. And we want to know if our data back that up. Well, first of all, we look at the means. The mean of the shady one is higher than the mean of the sunny one. But we also have to look at the error bars. And you can see here's an error bar right here. And here's an error bar right here. And what you want to look at, do those error bars overlap? Do they cross the same width in this case? And here they don't. And when your error bars don't overlap, that tells you that it's likely that the differences between your two sets of data are significant. They're statistically significant, which backs up our claim. We're saying that now we can claim with some certainty that plants grown in the shade have wider leaves than plants grown in the sun, at least for ivy. Now here's another example. So here we're looking at the average score of boys plotted against girls on a test. I can't read the y-axis here, but the boy average seems to be lower than the girl average. But notice that we've got error bars, and that notice that the girls had a lot of variation in their data. They've got a wide set of error bars. The boys were pretty precise. They had a lower average, but pretty precise. The error bars are small. But point being, the error bars overlap. Right in there, there's overlap between the error bars. And what that means is even though the sample mean here is lower than the sample mean here, the population means for the boys and the girls likely fall within the same range. There's a good chance that the population score for all the boys is the same as the population mean for all the girls. When error bars overlap, that tells you that more than likely there's no statistical difference between your two groups. When there's overlap, there's no statistical difference. When there is not overlap, like in this problem, there is a statistically significant difference. Now, that being said, we want to wrap up today with a couple of problems. So I want you guys to go ahead, first of all, and read for me what you see on the screen. So just read the whole problem real quick. All right, so we're essentially looking at how does the diet affect the change in mass of rabbits. And we're testing 3 different foods. We're giving some of the rabbits grass, some of the rabbits brand B food, and some brand C food. And this asks us to do a lot of things with the data. Calculate the mean, the standard deviation, and lots of other things. So let's go to the next screen. So here I have data set A, this set of data. And first of all, I added that data up and I divided by the number of data points, 6, to get a mean, remember, which is X bar of 7. And now I'm going to use that to calculate my standard deviation and my standard error. So first of all, I'll list all my individual data points in column A. So I had 4, 4, 5, 8, 10, and 11. Keeping in mind, this is my formula for standard deviation. I went ahead and made a column here for just this piece of the formula. So xi minus x bar. So in this case, that would be 4 minus the mean, which was 7, to get -3. 4 minus the mean of 7 to get -3. There's a typo. 5 minus the mean to get -2. 8 minus the mean to get 1. 10 minus the mean to get 3. And 11 minus the mean to get 4. All right. Now I went ahead and made a column now that takes into account this whole piece right here. So xi minus x bar squared. So I squared this column. So -3 squared to get 9. This should be -3 squared to get 9. -2 squared to get 4. 1 squared to get 1. 3 squared to get 9. And 4 squared to get 16. Now, to calculate my standard deviation, first of all, I want to sum up xi minus x bar squared. So add up this column. So let's see, that would be 18 plus 4, 22, plus 1, 23, plus 9, 32, plus 16 to get 48. So xi minus x bar squared, the sum of that is 48. I want to divide that by n minus 1. Remember, n is the size of your sample. I had 1, 2, 3, 4, 5, 6 in my sample. So n minus 1 would be 5. So I take the square root of that, 48 divided by 5. Take the square root of all of that to get 3.10. So that's my standard deviation. And then my standard error of the mean, and we got that formula down here. Standard error of the mean is equal to the standard deviation, 3.10, divided by the square root of the sample size, which is 6. So 3.1 divided by the square root of 6 to get 1.27. All right, so that's for sample A. Now I'm going to go ahead and here's my B sample. Let's make sure I didn't make any crazy typos on this one. So I added up my data, divided by 6 to get a mean of 11. Here I went 8 minus 11 to get -3, 8 minus 11 to get -3, 9 minus 11 to get -2, 11 minus 11 to get 0, 14 minus 11 to get 3, and 16 minus 11 to get 5. Those look good. I square them to fill in this column, sum them up, divide by 6 minus 1, 5, end up with a standard deviation for this group of 3.43. Divide that by the square root of 6 to get a standard error of the mean of 1.367. All right, so notice that the standard deviation in this group is a little bit bigger than it was in group A. And that just means the data is a little bit more varied in group B than it was in group A. Here's the same scenario for C. Here are my data points. Add them up, divide by 6 to get a mean of 5. 2 minus 5 to get -3. 4 minus 5 to get -1. Same here. 5 minus 5 to get 0. 6 minus 5 to get 1. and 9 minus 5 to get 4. Square them, add up those sums, divide by 6 minus 1, n minus 1, to get a standard deviation of 2.366. And then divide that by the square root of 6 to get 0.966. Again, notice lower standard deviation, which means these data are closer together than are these data and these data. That being said, now we want to draw a graph. And this asks us to graph. Let's go back to our question. This is really the important part. So we calculated the mean. We calculated the standard deviation. The largest standard deviation was in data set B, 3.347. And remember, standard deviations tell us about the variation within the data. We calculated the standard error of the mean for each group. Now, the important part, F. Graph the means for each treatment group using a bar graph. Use inferential error bars based on SEM, standard error of the mean, to depict 95% confidence intervals for each data set. So let's think about what our means were. So for A, we had a mean of 7. For B, we had a mean of 11. And for C, we had a mean of 5. So first of all, these are going to be our mean change in mass. So we want to label our axis. You want to put units, so those changes were in grams. You always put your dependent variable on the y-axis. And our independent variable in this case is just going to be the group, the kind of food that we used. So I'm just going to spread these out. So this is we'll just call these our treatment groups. And we had treatment A, treatment B, and treatment C. And we're going to graph those means. So let me remind myself what the means were. So 7, 11, and 5. So 1, 2, 3, 4, 5, 6, 7, 8. So I need to make each one of these be 2, I think. So you've got to determine how to set off your axes. So my first mean for group A was at 7. So I'm just going to draw a little bar here at 7 like so. Second group was at 11. Like so. And third group was at 5? Yes, 5. So like so. Now we've got to draw our bars, our error bars. And remember that to do those, we're going to go up and down from these sample means, which I'm putting a dot at the sample means. We're going to go up and down from those 2 standard errors of the mean. So for group A, our standard error of the mean was 1.27. We want to go up from there 2.54. So our mean was at 7. We're going to go up 2.54 from that. So that would put us up at about 9.54. And I'm going to go ahead and write that in. You don't have to, but that just tells me where it's at. So that's my first error bar. And I connect that down to my mean with a vertical bar like so. Now I also want to go down 2 standard errors of the mean. So down 2.54. So I'm going to go 7 minus 2.54 to get 4.46. So that would be somewhere right in here. Connect that to my mean. So what that's telling us for group A, for sample A, my sample mean was at 7, but there's a 95% chance that the population mean for all of the grass-fed rabbits is going to be somewhere between 9.54 and 4.46. Let's do the same thing for group B. For group B, our standard error was 1.367. So I want to multiply that by 2 because I want to go up and down 2 of those. So I'm just going to write this number over here so I don't forget it. I'm just going to round it to 3.37. That's 2 times the standard error of the mean. So I need to go up from the mean that far. So I'm at 11. So that's going to put me at 14.37, somewhere right in here. Connect that to my mean. And then I want to go 11 minus 2 standard errors. So 11 minus 3.37. That puts me at 7.63. So that's going to put me down in here somewhere. So that's saying even though that my mean for this group, my sample mean was 11, there's a 95% chance that for all of the rabbits fed food B, that the population mean is going to be between 14.37 and 7.63. Finally, for C, our standard error is 0.966. We're going to multiply 0.966 by 2 to get 2 standard errors. That's going to give us 1.93. We want to go up 2 of those from our sample mean. So our sample mean was 5. So that's going to put us at 6.93, somewhere right in here. Connect that to our mean. We also want to go down 2 of those. So 5 minus 1.93 puts us at 3.07. So somewhere right in here. All right, so what I'm hoping that you see is that sample C had the lowest mean mass gain. Sample A had the next lowest mean mass gain. Sample B had the highest mass gain. But what I'm also hoping that you see is that these 2 overlap. What that's saying is that there's probably not a significant difference between the mass gains between A and C. So we're saying that the grass, even though the sample mean was a little bit higher, it's probably not any better at causing the rabbits to gain mass than food C was. Notice that also A and B overlap right here. So there's no statistically significant difference between the mass gains of rabbits fed A than those fed B. Really, the only place where there's a significant difference is between B and C. And notice that there's no overlap between the error bars for B and C. So B, food B, causes rabbits to gain weight better than food C does. It's a statistically significant difference, more than likely. So let's go back to the question. So based on the sample means and the SEMs, are the mean changes in mass amongst the 3 groups statistically significant? The only place where there's statistically significant differences are between groups B and C. And your justification would be that's the only case where the error bars or the confidence intervals don't overlap. And that's really all you need to say for justification. B and C are statistically different because their error bars or their confidence intervals don't overlap. And that's enough to get credit. All right, let's wind this up with one more quick question. So I want everybody to take a minute and read question 8. And we'll end this here in just a second. So go ahead and read 8. And go ahead and look at parts B, A, and B, and come up with an answer in your head. Just formulate an answer in your head, and we'll talk about it here in just a second. All right, so the researchers think that their hypothesis here is that exercise is going to cause males to make more prolactin. Prolactin is a hormone that eventually causes males to also make more testosterone. So essentially the idea is more exercise and more testosterone is really kind of where we're going. But in this case, we're looking at just prolactin. So here's the males that exercised. And you look, the sample mean of the males that exercised, after 1 hour, they had a higher prolactin than they did an hour before. But notice the error bars overlap, which means there's really no statistically significant difference between before they exercised and an hour after they exercised. And then without exercise, here's where the males without exercise started as far as prolactin levels go. Here's what they had an hour later. And again, note, error bars overlap. Also note that there's overlap between the no exercise group and the exercise group. They're all overlapping. So A, justify the use of the without exercise treatment as the control in the study design. Well, our independent variable in this case was exercise. And our dependent variable was prolactin levels. And we're essentially trying to figure out, does exercise have an effect on prolactin levels? So a perfect negative control is no exercise. When you're creating a negative control group, essentially you want everything about your subjects and treatments to be the same except for the independent variable. And typically in a negative control group, you don't expose the members of the control group to the independent variable. So this makes perfect sense. So essentially, the no exercise group is a standard negative control. You're not exposing the subjects to the independent variable, which in this case is exercise. B, using evidence from the treatments, determine whether prolactin release changes after exercise. And really, the point would be, here's where we didn't exercise. Here's an hour after. And even though it looks like exercise, that the prolactin levels go up after an hour, the error bars overlap. And because the error bars overlap, there's no statistically significant difference between our prolactin levels before the exercise and an hour after. So our answer would be the exercise has no statistically significant effect on prolactin levels because the error bars or the confidence intervals for the with exercise and without exercise groups overlap. That's the justification. All right, there's one more quick thing I want to look at, guys, and I thank you guys for staying over for a couple of minutes. Here's a quick problem, and this is the kind of thing, this is straight off an AP exam a few years back. And I'll just walk you through this real fast because this is a different kind of error bar question than what we've been looking at. So here we have some male guppies, a kind of fish, and we're looking at how many spots they have. So this is telling us that adult male guppies have spots, while juvenile and adult female guppies don't. So in a study of selection, male and female guppies from genetically diverse populations were collected from different streams and placed in an isolated environment containing no predators. The study population was maintained for several generations in an isolated area before being separated into two groups. One group was moved to a pond with a predator. One group was moved to a pond with no predators. The two groups went through several generations in the new environments. At different times during the experiment, the mean number of spots per adult male guppy was determined and shown in the figure below. Vertical bars in the figure represent two standard errors of the mean. So we've got error bars. So first of all, question A, describe the change in genetic variation in the population from 0 to 6 months and provide reasoning for your description based on the means and the SEM. So change in variation. Remember that standard deviation tells us variation, and that standard error of the mean is equal to the standard deviation divided by the square root of the sample size. So really, standard error of the mean also looks at variation. Greater standard error means more variation. It wants us to compare 0 to 6 months. So here's at time 0. And I want you to notice that the error bars are really wide. When you have wide error bars, that means lots of variation. But notice that by time 6 months, right here, notice the error bars, very small. So narrow error bars mean little variation. So what they wanted you to answer here on part A is that from 0 to 6 months, the amount of genetic variation decreases dramatically. And you know that because the error bars at 6 months are much smaller than the error bars at time 0. That's your justification. You could say that your confidence intervals, your 95% confidence intervals at 6 months are much smaller than they are at time 0. That's what they want you to answer on part A. Now I'm going to end up with parts B and C, review a little bit of evolution for you, and then we'll call it a day. Propose one type of mating behavior that could have resulted in the observed change in the number of spots per adult male guppy between 6 and 20 months in the absence of a predator. So here's 6 months. 20 months is over here somewhere. We're looking at absence of a predator. So we're looking at this line, predator absence. And notice that the number of spots went up. Well, there's no predator. So what you got to think about, well, why would the spots go up when there's no predator? And the idea here is that spots are used as a way for the males to attract females. The females are attracted to males with lots of spots. This is what we call sexual selection. Males with more spots are selected by females, and therefore they have more offspring. So your answer to B, the type of mating behavior is sexual selection. And then on C, propose an evolutionary mechanism that explains the change in average spots from 0 to 20 months in the presence of a predator. Well, that's this graph. And notice that when a predator is present, the number of spots drops. And the mechanism here is natural selection. Because what's happening in this case is that males with lots of spots are being seen by predators. And because they're seen, they get eaten more. So males with more spots show up not only to the females more, but also to the predators more, and they're eaten, and therefore they don't get a chance to reproduce. So the number of spots drops from generation to generation. All right, that's it for today. We're way over time, so we're going to call it a day there. Thank you guys for being here, and I hope to see you again next week. Sorry we got in such a rush today. Thanks for being here. Hope it helped.